It is indeed my pleasure to introduce Dr. Suzanne George, who's joining us today from Dana-Farber Cancer Institute in Boston. What an impressive resume Dr. George has. She's originally from Boston and graduated magna cum laude from Harvard. In 1995, she received her MD, followed by 6 more years of residency and fellowship in internal medicine and hematology oncology. Dr. George's 14-page vita outlines all of her many clinical trials, publications, speeches, and presentations which have become a part of what she offers to the GIST community. Her research focuses on clinical trials and outcomes in soft tissue sarcoma such as GIST. She has developed several phase 1 and 2 institutional protocols in addition to developing and executing multi-center trials. She is also currently a co-investigator in 28 phase 1, 2, and 3 trials. She's a co-investigator with DFCI Cancer Genetics team on multi-center study, which is evaluating genetic factors which may impact the development of GIST. Her role is the lead medical oncologist on this particular team. This study may one day help us answer the questions we all ask: why and how? Working with surgical colleagues, Dr. George developed the first institutional tissue repository with clinical annotation for patients with soft tissue sarcomas. This initiative provides an essential resource for clinical, translational, and outcomes research in the field. Dr. George's clinical focus is treatment of patients with sarcomas, including GIST. She works collaboratively with specialists and develops treatment plans for all of her patients. She is widely sought as a national and international speaker on a variety of subjects dealing with GIST therapies, treatments, and research. Thank you, Dr. George, for all your special efforts, and we welcome you to Houston.
Well, thank you so much for inviting me. I really want to thank Dr. Trent and Barbara and the organizing committee, and I'm so happy to be here and just really so impressed at your interest and enthusiasm, and really want to help to provide information whatever way can be most helpful for you. As was mentioned, I'm from Dana-Farber. I'm a medical oncologist there. I work with George Dimitri. He's our leader, and I'm very lucky to have a great team and have been part of that team for some time now. My talk today is focusing on the emerging role of imatinib blood level testing in advanced GIST. I also am involved with Project FLAG, which you may know about from the website, and that's the genetics issue that was mentioned, and I'm happy to talk about that as well if you guys have questions at the end or if at any other point. So I included my mandatory disclosure, which I include for all my talks, which is just that I've served for advisory boards for both Pfizer and Novartis. And I just want to start by saying, gosh, we've learned so much about GIST in the past decade. We've learned a lot about the importance of KIT as an important signaling pathway in GIST and an important target in therapy. The application of tyrosine kinase inhibition, meaning blocking KIT and other proteins that are similar to KIT that are thought to be important in GIST development. We also are understanding that unfortunately that resistance does develop to this class of drugs for many patients, and we need to understand why that's happening and how we can get better strategies to overcome it. And in that vein, there are multiple ongoing efforts to try to develop new therapeutic strategies, both within new drugs, applications of drugs in new ways, and new integration of surgery and medicine to try to optimize therapy for patients with GIST as well as to combat resistance. So this is a slide that I used to use a long time ago when I first started giving GIST talks, and then I stopped, but I think it's relevant now, and I've sort of brought it back out again. This data comes from a retrospective study in Sweden that looked at 600 patients tumors that would currently be classified as GIST by current modern day criteria. But if you look historically, prior to the year 2000, these things GIST was called by all different kinds of names. Only about less than a third of the time was GIST actually given the right label. And it was because of that lack of ability to classify the tumor that we were really not able to study the tumor so well with regard to standardized approaches to treatment. And it's because we were studying all different kinds of things and we didn't actually recognize that this was one entity. And things really changed in the late 90s and 2000. This was a paper that was published by a team in Japan that really identified the importance of the biology of GIST and saying, gee, in GIST, we find gain-of-function mutations in this protein called c-KIT that leads to the tumor growing and dividing. And what that meant was, this is a schematic that just says, here's KIT, the KIT receptor. It sits, let me see if I have my arrow. It sits across the cell membrane. And typically, it would just sit there quietly unless there was something out here to turn it on. But in normal cells, usually it's not turned on. It's just kind of sitting there. But what happens in GIST in most cases is there's a mutation here that causes this tyrosine kinase and this tyrosine kinase to come together and turn itself on with activation of ATP, which is sort of the energy signal, into the kinase domain, and that this activation with ATP leads to phosphorylation or turning on the tumor cell. And when the tumor cell gets turned on, it basically switches on all these pathways downstream that says, okay, even though I don't have anything turning me on here, this mutation is going to act like that switch. I'm going to turn on and I'm going to grow and I'm going to divide, and I don't have good things to control me moving forward. And it's really the identification of this mutation in tyrosine kinases, primarily KIT, that has led to our understanding of GIST, that's led to our ability to diagnose GIST more as a disease entity, and to help have us optimize therapy. So how does imatinib work in relation to this scheme? Well, imatinib or Gleevec works by binding that area where the phosphorylation occurs, where that red P happened that turned everything on. If that ATP, that energy source, cannot get in there, then everything downstream doesn't flow. So imatinib gets in there and sort of jams that signal up so that things cannot flow downstream. And so that although there's a mutation that's saying, okay, I'm going to turn on KIT and I'm going to keep going, imatinib gets in there, blocks it, and stops proliferation and survival of cells that are driven by this mechanism. And what we know is that's how it works in the lab, and we know that clinically it made a huge difference in the introduction of imatinib in advanced GIST has made a huge difference with our ability to treat patients with GIST and control disease in GIST. And this is a summary slide from B2222, which was the original randomized phase 2 trial using imatinib at either 400 or 600 milligrams per day in patients with advanced GIST that had not been treated with tyrosine kinases before. And what we've seen is that over time, only about 10%, so like close to 90% of patients achieved disease control with either cessation of growth, so the tumor stopped growing, or significant shrinkage when imatinib is used. And we think that the reason for that is because imatinib is jamming that pocket so that those signals that were basically turned on now cannot flow. And not only is imatinib leading to disease control and to tumor shrinkage in some cases, but it's clear that the introduction of imatinib and tyrosine kinase inhibition inhibitors are leading to patients living longer and living better. But what's also important to note is that as we're learning over the years, not all GISTs are the same. In my initial schema, I described this activating mutation, that red star that sort of turned things on. But we know that activating mutation is not the same in all GISTs. About 87, so close to 90% of tumors of GISTs carry this mutation, but the majority of them seem to occur right here in what's called exon 11. A smaller percentage occur in exon 9, and then even a smaller percentage occur in exon 13 or exon 17. Now, some patients actually have a completely normal kit, and they don't have any of these mutations present, but they do have some mutations in a sort of a sister kinase, which is platelet-derived growth factor receptor alpha, and those are relatively low frequency, but they can occur.